Okay, thank you, Camille. Hello, everyone. My name is Llusia. I work at the University Autónoma de Madrid here in Spain. And first of all, I would like to thank all the organizers for inviting me to this interesting meeting. Part of my work focus on developing and applying these techniques, passive acoustic monitoring. I'm a big fan of this technique, and I thought that at the beginning, maybe we can try to answer this question, why maybe this new tool could be a good solution for monitoring biodiversity and ecosystems. Probably in the audience, there are people just starting to getting interested in this method. So I list a little of reasons, a list of characteristics that make this method, I think, interesting alternative or complementary method for monitoring. And then in this talk, I will move for the core question of the presentation, which is how can take advantages of this type of tools to make better studies. How can we develop our acoustic programs in terms of sampling, and talk a little bit about spatial and temporal sampling. So first of all, I think this acoustic monitoring is a versatile method, as we can, as Luis commented just before, we can monitor very different animal groups that use acoustic communication, and then we can measure different ecological behavioral traits from the information we get with acoustic sensors. But nowadays also we can analyze the sounds in a more general way using, for instance, unsupervised analysis of what we call soundscapes. For instance, there's new acoustic diversity indices, which are growing in the last years. So finally, we can measure and apply this method for a wide range of research topics. Also, I think it's interesting because it's quite, I can say, easy to apply in some way. I think in terms of the protocol, the technology is not that complicated, and nowadays we have plenty of different acoustic sensors available in the market for applying and for using this technique. When I started to do my PhD, we needed to build our own recorders. Nowadays, it's much more easier to use this. Also, there is a lot of benefits in terms of sampling when we use acoustic automatic sensors, especially because we can increase our sampling effort and then increase the detectability in our studies. So we can record in many different places simultaneously on the long term, which is obviously quite interesting. But there is a question also, a key question that we need to answer is this method effective when we monitor animal communities? Well, there is many papers trying to deal with this key question, and recently last year we published a paper when we compare traditional surveys after searches with passive acoustic monitoring, and we wanted to do this in a very challenging and complex environment like tropical savanna in Brazil. There we did after searches and passive monitoring at the same time in a series of annular assemblages with many different species. And what we found was that when we compare sampling in different scenarios, first we found that species reasons were similar when sampling was biased towards the traditional survey. However, when we increase sampling and we equal sampling effort conducted by the two different methods, as you can see in the figure, we could identify a higher number of species when we use acoustic monitoring. And this was even higher when we increase sampling with this acoustic sensor, which is obviously a key characteristic of this tool. Also, all the species we found with the traditional method, we identify also with acoustic sensor. So I think we can conclude that this method is quite effective. So now we can move then to try to identify what kind of designs in terms of spatial, but also in time, we need to apply for taking advantages of this technique. So for that, we thought that the first thing we maybe can explore is how the people are using this method, because we can probably learn some lessons from the literature. Maybe we don't need to copy what other people are doing, but maybe it could be a good start, and we can get ideas from that. So during the PhD of Larissa Sugai, she reviewed almost, I think it was like 460 papers published during 3 decades, using these techniques in terrestrial ecosystems. The first paper we published was in Bioscience a few years ago, and the first thing we identified is, as Camille said before, this is an emerging technique. As we can see from the literature, there are increasing number of papers using these acoustic sensors. But also we identified 2 features that I think are relevant. First of all, as we are saying, papers are using this technique for many different groups of animals, mainly bats and birds, but also anurans and others. And for asking, exploring different type of research questions, a wide range of topics. So here is also, we can wonder if this method is so wide in terms of taxa and topics, is possible to define a standardized method for being applied in all these different situations. So the next step was trying to explore more about the specific design that these studies are applied. And for progressing in the research, we analyze more about these papers with the collaboration of Camille and Donquet, and we publish another paper in a few recently also. First, talking about the spatial design, we identified that most of the studies were conducted on macro scale, saying that the study sites were placed more than 20 kilometers away one from the other one. In general, the number of sites were highly variable in the studies, but what we see here is that usually this technique allows us to conduct the studies at large scales. Also, was quite surprising seeing that most of the research was conducted using low or intermediate number of recorders, between or less than 11 recorders, I would say. So I think probably we are seeing now an increasing trend in the number of recorded uses because nowadays we have more options and low cost sensor in the market. So probably in the future we will see this trend, this pattern change. But in general, people are using not a lot of recorders, but only low or intermediate numbers of them. This has particular implications in terms of the spatial resolution of the data, but also in terms of storage and the type of analysis we need to conduct. Also, we explore the sensor arrangement, how is the spatial configuration of micro scale of the sensor. And we found that most of the study choose single recording station, followed by random configuration transits and grids that were in general less common. So apparently, independent of the spatial distribution of the study species, most of the study prefer to use a single point station as their study. Then another decision that we can make is if we let the recorder fixed in those points, or we move the sensors between study sites. As you can see in the figure, I was quite surprising. Rotating the recorder between sites was a common strategy, probably for increasing the number of sites when the people have low number of recorders. So if you have less number of sensors, a solution could be move them between sites to increase your sampling. But this is also related with this number of samples. As you can see in this figure, when the research team had more sensors available, they prefer to use fixed recorders instead of rotating them. Why? Because probably this allows us to conduct simultaneous sampling at longer periods. And finally, most of them prefer to use recorder or keep the recorder static during the recording session instead of moving, like travel with the recording along the person. Well, in general, for making a good design, I think, as Luis also commented before, I think we need to explore a little bit more about what is the detection space of the sensor, which is not that easy, but quite relevant to try to determine which is the better position of the recording, the distance between them, and also for determine the area in which we are monitoring, and then extrapolate this for using or for estimating abundance or density. There are several papers that design methods to determine this detection space, but usually this is not very, I mean, people are not using these techniques often, mostly because are quite complex in terms of the method. So in general, what we are seeing is that probably in the future, the number of recorders available for those programs are going to increase. Then probably we can use them in another different configuration, like grids, transit, that nowadays are less common. But in general, researchers prefer to use this at micro scale with fixed and static sensors. And then here a few questions that I think are relevant in terms of spatial design. That how can we apply easier method for estimating detection space, or how can we standardize these decisions for making more comparable data across studies. In terms of temporal sampling design, we identified key differences in the number of studies using monitoring during specific periods of the time of the day in comparison with studies recording during the 24 hours. As you can see, most of the researchers use continuous recording, but in specific periods of the day. When they record during the entire 24 hours, discontinuous recording were more common. Obviously, streamlined periods are more common than others because most of the animals are active at these periods. And when we look at the specific recording schedule, they were quite diverse, as you can see from these figures, that show the number of recordings per hour and the recording length. So there were many different types of schedule that usually there are obviously a negative association between the number of recordings and the length of those files, as is very obvious. And this is important because our temporal resolution of the data in the observation is dependent on these patterns. So usually I think it's advised to have shorter time windows across your period of sampling for getting a better temporal resolution in your data. But this is obviously also dependent on your capacity for storage and the type of analysis that you want to apply them. So in general, probably in the future, with improved capacity for analysis and storage, we will see more programs using 24-hour recording and continuous recording. Although I think that regular sampling, sub-sampling will be still needed in the future. Open question, how can we apply methods for better estimating species detectability and then use this information for improve our temporal design? Or how can we standardize this decision and sampling? And just briefly to show what we are doing right now using these lessons and this experience we get from this is monitoring a series of sites in Brazil. We developed a monitoring network all around the country thanks to many different institutions and colleagues that are recording there. And we had to make those decisions and try to build a network. Finally, what we decided was, like, in this case, we are recording mostly Anuran communities. So we set a single point station, fixed and static, recording 24 hours, that using regular sampling for improving our capacity for storage and analyzing those data. So now we have approximately between 1 and 2 years of recording continuously in those study sites. If you want to take a look more about all these protocols, you can take a look in the paper. We provide some guidelines about this and also key literature related to these methods. Just I want to thank all the institutions that are helping and funding on this project, all the colleagues, and well, saying that we have now open positions related to this research project. So if you are interested in doing maybe a PhD or postdoc and you want to join us in the lab, you can contact me. That's all. Thank you so much for your attention. I think I'm fantastic.
Yeah, perfect. Thank you, Diego. Thank you very much.
You're welcome.
So if you have questions in the audience, please raise your hand. Or if you have questions here, feel free to ask them in the room. And I see there is a new message. Someone says, "Hi, Diego." Rodrigo, now I don't know how to
Okay.
if I pronounce.
I can start up if there is no rising hand so far.
No, you can have
Yeah.
you can ask the question if you want.
Okay, thank you very much to both of you. Those were very useful and you talks. And one question for Diego, just in the first comment. It's such a pleasure to see that bioacoustic is displayed for the amphibian inventories in Brazil. That is, we all know the most treated group of vertebrates. So we see all the encouragement for that. And one question, very short. Maybe a misunderstanding of two of your slides.
Sure.
About the daily recording. Since your previous slides were showing, that's the number of articles.
Yeah, that's the number of articles depending on when they are recorded.
When they are recording, and then the other one,
we focus on dial periods, those studies that are recording only in particular periods of the day, right? When they are recording in particular. So as you see, there are it's more common that people record at night, right?
Yeah.
During sunset, night, and also during dawn, than in the middle of the day, which is, I mean, quite obvious in terms of the activity of birds, animal insects, and so on. So this figure is representing like an histogram, right? Presenting the percentage of studies recording a particular period of the day.
All right, and then the next one is
And
about
then
the richness.
No, this one is showing for those studies that are not recording continuously, but recording at a particular with a particular sampling, regular sampling, right? Which is the number of recordings in an hour. For instance, you can see that in the bottom, you find like between 1 recordings to 6 at maximum, right? Only a few papers use 10 files or record 10 files in an hour. And in the vertical axis, you have the length of the recording, how the number of minutes that these recordings are working.
All right, thank you very much for this.
But the x-axis is design, or is the number of observations you make because you...
No, it's the design. The side of the circles are the number of the studies. So in general, you see this negative relationship because usually if you have many different files, you probably choose that those files are shorter, right? And if you, for instance, the blue circle at the top means a single recorder during 30 minutes. So that means a recording is scheduled 30 minutes on, 30 minutes off, right? So my experience, I think, is more interesting to split that effort in many different time windows for getting a better data resolution. But this recording length will depend on the activity of the species. If we are working on particular group of animals, this is depending on which is the interval of the signals the animal are broadcasting, right? So if we are monitoring a particular species that just produce sounds every, I don't know, 1 minute, maybe we need a large time window to catch, I don't know, a few signals, right? But if the animal is continuously singing, probably we can shorten the recording and place different files across the hour, improve our temporal resolution of the data.
General, these studies focus on activity or assessing the number of individuals.
These are, I mean, super wide. I mean, most of them, but activity patterns, but also behavior, community composition, and so on. So what we are seeing from here is that we still need some standard protocols to decide how we determine this sampling, because usually people are applying their own experience about the study group and so on. So there are still work to be done up here for determining a little bit more clearly how can we decide about this key element in acoustic sensor, which is the programming of the sampling, the temporal sampling. Yeah. So there are much debate, I think, on this.
So in the chat, I saw one comment which is not directly related to your talk, which is about so Kali Batis from Conservation Tech Directorate says on their website they have a compilation of resources, organization, companies, and projects in the conservation tech and tech for wildlife space. So if you have something you want to share in there, or if you want to just check the directory, you should do so. Then there was Eugenia Degano asking, she says she's new to the field and she's wondering what acoustic recorders are recommended for this. Because a lot of studies use SM4, but it's expensive. So, and Audio some right now they have supply problems. So she is asking whether anyone has tried the SM micro. I don't know if you have a personal experience.
Yeah, I mean, I think there are others too. Most of the recorders in the market, I think, are proper. They provide different characteristics, as he said. So most of them are fine. So depending on if you want a sensor with many different settings that is going to last during years and under different very difficult conditions, maybe you need to spend a little bit more money on this sensor. But if you want to spread many numbers of recordings in your study area, maybe you need to buy cheapest one to increase the number. So I mean, this is very specific. Also regarding your question, that if you want to write us, we can provide more information about this issue.
And there are a few people also responding saying that they have different experience with it. So I think they will have plenty of feedback.